Hi everyone, my name is Joana Loureiro. I'm Associate Principal Scientist at Stemmatters, and I'm here today to tell you about human trophoblast stem cells derived induced pluripotent stem cells, a scalable pluripotent platform for regenerative medicine applications. In this slide is a quick overview of what I will be covering today. I will start by introducing who we are and outlining our product verticals. From there, I will walk through stem cells, human-induced pluripotent stem cells, iPSCs, and human trophoblast stem cells, hTSCs. We will also look at the current landscape of the iPSCs clinical trials. Then, I will share details on our approach of generating, expanding, and banking GMP-compliant iPSCs, along with the respective quality control. And in the end, I will show our strategy and results for the expansion and banking of the accelerated bioscience hTSC-derived iPSCs. Stemmatters is a biotechnology company based in Portugal that operates as a contract development and manufacturing organization, so it's a CDMO. Stemmatters was founded as a spin-off from University of Minho. Initially was focused on stem cells and tissue engineering, and in 2014 the company was approved as an European Union tissue establishment for isolation, processing, preservation, and storage of mesenchymal stem cells, or MSCs. This milestone not only increased the company's technical expertise with human MSCs, but also strengthens our GMP capabilities, so good manufacturing practice, and regulatory knowledge. Since then, the company has gained extensive scientific and technological expertise in a variety of regenerative medicine segments, including hands-on experience with blood-derived biologics and other cell types. We address clinical manufacturing challenges across three key verticals: advanced therapies, complex biologics, and raw materials. On raw material side, we specialize in the scalable production of human platelet lysate for use in culture media. On complex biologics, we are active in the manufacturing of blood-derived biologics conditioning media and are taking the initial steps in the small-scale exosome production. In advanced therapies, beyond our experience with MSCs, we support the clinical translation of innovative therapies in two strategic areas: the cell-based immunotherapies that include dendritic cells, T cells, CAR-T, and CAR-NK cells, and pluripotent stem cell technologies. The last, through a collaboration with Accelerated Biosciences, to bring their proprietary hTSC platform to the European Union market. Before I move on to hTSC-derived iPSCs, I would like to give you a brief overview of stem cells, what they are, and where they naturally occur. So, stem cells are undifferentiated cells with two key properties. First, they can replicate themselves, a process that is known as self-renewal, and second, they can differentiate into specialized cell types. Regarding the types of stem cells during the embryonic development, as you can see here in this slide, we first encounter the totipotent stem cells. And these cells are the more versatile cell type since they can have the unique ability to give rise to all cell types. As development progresses, cells become more specialized. And in the blastocyst stage, in the inner mass of the blastocyst, we can find the embryonic stem cells. These cells are pluripotent stem cells as they can give rise to the three germ layers: the endoderm, mesoderm, and ectoderm. These germ layers appear later in the gastrulation phase of the embryogenesis. These three germ layers are the foundation for all tissues of the body, and this feature is what makes pluripotent stem cells so valuable for regenerative medicine. Additionally, the trophoectoderm layer of the blastocyst, we can find there the trophoblast stem cells, and these cells are early stem cells that are responsible for forming the extraembryonic tissues, such as the placenta. Pluripotent stem cells, because of their ability to give rise to all the cells of an adult individual, hold an extraordinary promise in biomedical research and regenerative medicine. Although pluripotent stem cells are naturally present only during early embryonic development, technological advances have made it possible to generate them from somatic cells taken from adults. So, easily accessible cells, such as fibroblast blood cells, can be isolated from adult individuals, cultured in laboratory, And then, by introducing specific transcription factors known as reprogramming factors, as is the case of YAMA factors, SOX2, OX4, PLF4, and CMYC, it is possible to remodel the chromatin and gene expression of somatic cells and effectively return them to the pluripotent state. As a result, scientists can create an unlimited source of material for experimental and therapeutic applications, and these cells are particularly promised in the field of cell therapy since they can provide access to key human cell types that may be used in cell replacement therapies for both autologous and allogenic applications. Some examples of these difficult to obtain target cells include the insulin-producing cells, cardiomyocytes, and neuronal cells. In a recent review on the status of human pluripotent clinical trials worldwide, we can see a cumulative number of trials initiating each year, and this has grown exponentially since 2010. Early on, there was an exclusive focus in embryonic stem cells, here represented in green, especially in the first few years, and the first clinical trial using iPSCs appeared in 2013. Followed by a more consistent use in 2017. Since 2020, iPSCs used in allogenic therapies have accounted for the large share of all trials, as we can see in yellow here in this graph. This review also shows that most of these studies remain in early phase of clinical trials. The majority are either in phase 1 or phase 1-2 trials, with only 1% in phase 3. In terms of therapeutic areas, the immune blood products dominate the clinical landscape. However, stromal products, ocular therapies, and central nervous system applications are also well represented in this graph. Finally, if we look to the world map, we can see that most of clinical trials are concentrated in the United States and in China. As overviewed in the beginning of this presentation, the human embryonic blastocyst is composed by two main cell types, the embryonic stem cell and trophoblast stem cells. These cells, or hTSCs, can also be obtained from preplacental tissue chorionic violet in early pregnancy stages. And these cells, the hTSCs, represent a promising starting material for development of iPSC-based therapies, offering several important advantages, such as they are early aesthetically sourced stem cells derived from embryos, have a higher reprogramming success rate comparing to adult stem cells due to their epigenetic features that closely resemble the early embryonic state, as well as their high proliferative capacity. Compared to the other cell types, the hTSCs have a lower burden of post-zygotic mutations due to reduced number of DNA replications and DNA damage, as well as low exposure to environmental damage when compared to adult somatic cells. And developmentally, they are closer to embryonic stem cells, which further support their use in reprogramming. Accelerative Bioscience has a patented hTSC platform that uses plurimplantation human trophoblast stem cells derived from ectopic pregnancies, and because of this hTSC's remarkable feature, Accelerative Bioscience uses this cell as a foundation material to reprogram into iPSCs to create a reliable source material to support the development and cell therapies innovative. To generate iPSCs under GMP-compliant, so good manufacturing practices, the workflow begins with the collection of the donor primary tissue. Once the tissue is received, the process continues in the GMP suite with isolation and expansion of primary cells to create the primary cell stock. After primary cell stock has been established, the cells are reprogrammed using reprogramming transcription factors as described in the previous slide. And over time, the iPSC colonies will start to emerge. Then the iPSC clones are individually expanded to establish the iPSC seed stock. Finally, vials from this iPSC seed stock are used to create first master cell banks and later working cell banks. For generation of a GMP iPSC cell bank, there are key points to consider, namely the donor eligibility. In the European Union, it must follow a specific regulation, that is the Regulation 2024/1938, that was approved last year, and this regulation binds the rules for the donation, testing, processing, and etc. for any human-derived substance, namely cells and tissues. Xeno-free and serum-free media, substrate and reagents should be used in primary cell isolation and expansion, as well as in the other expansion steps of this workflow. The reprogramming method used to derive iPSC should not lead to the genomic integration to avoid insertional mutagenesis and should not leave residual vectors in the final product. And then, based on PC results, the iPSC clones are selected to establish master cell banks. Regarding the quality control tests to perform over this workflow, these are performed on primary cell stock with sterility, mycoplasma, endotoxins, cell viability, cell-specific markers, and adventitious virus. The adventitious virus testing will depend on the risk assessment results, considering the animal and human origin of components of cell culture, substrate, and other reagents that are in contact in the target cells. On iPSC stock, master cell bank, and working cell banks, the same QC tests are carried out with addition of karyotype, pluripotency markers expression, and trilineage differentiation of iPSC cells. This GMP process demands qualified or validated tissue sourcing, cell banking processes, standard operated procedures, form records, raw materials, operators training, GMP facility, and quality control analytical tests. The GMP-compliant process for iPSC banking under European Union regulation includes 3 sets of runs. All of these runs are performed or should be performed in the GMP suite, where the generation of master cell banks and later working cell banks are in the last set of runs. The first runs are included in the aseptic processing validation, or APV. These are needed field runs that consist in simulation of the process steps with higher risk of contamination. For example, the opening handling of tubes and opening handling of other flasks, to demonstrate the ability to implement the process under aseptic condition. In these simulation runs, instead of using the cells and respective growth media, we use this specific microbiological growth media that if any contamination occurs during this simulation, this growth media will easily detect the contamination. This APV requires 3 consecutive successful runs, as well as a periodic revalidation every 6 months. The second set of runs are the engineering runs, that are qualification runs, and these are full-scale tests of the manufacturing process. In the end, the product will not be released in case of these runs have the actual cells, media, and personnel. These qualification runs will allow demonstration of process robustness at scale and qualification of operators and equipment. There is not a defined number of engineering runs. The required number depends on the process and must be sufficient to demonstrate the GMP readiness of the process. The last run is the master cell bank and working cell bank establishment that includes the manufacturing process, QC tests of the product, and in the end, the release by a qualified person. So the release of the product by a qualified person. The GMP-compliant cell manufacturing requires documentation, SOPs, batch records, equipment calibration logs, deviations, and auto-specifications procedures. Personnel must be qualified, and trained staff must follow gowning procedures and have regular retraining. And regarding the facilities and equipment, there is the need to have qualified rooms and equipment, environmental monitoring, and change control procedures. After cell expansion and storage, the cells are subjected to a quality control panel for characterization of the following attributes: safety and purity, identity, and potency. For safety and purity, sterility, mycoplasma, and adventitious virus are tested, and the result must be negative. The adventitious viral panel is defined based on the risk assessment of the process, as explained in the previous slide. For endotoxins, the defined assessment criteria is less than 0.5 endotoxin units per ml, and the bank cells must have a viability rate higher than 60%. The genomic analysis must result in the normal karyotype without chromosomal or copy number variation aberrations. And regarding karyotype, the traditional G banding method is the method that is well known and used for quality control, but it has a low resolution between 5 and 10 megabytes, and other methodology allowing the detection of abnormalities in a smaller regions should also be considered. Furthermore, next generation sequencing or NGS should also be performed to confirm the absence of residual reprogramming vectors in the end products if applicable, since it depends on the reprogramming method. For cell identity, iPSCs must show more than 70% of the cells co-expressing at least 2 pluripotency markers, and STR profile of the cells must match the profile of primary cells and seed stock cells. Potency of the cells must be confirmed through the demonstration of cells from the 3 embryonic germ layers, and then, in the case of the master cell bank met all the acceptance criteria, this master cell bank or working cell bank can be released. The stability of the master cell bank and working cell bank needs to be periodically assessed using a defined QC panel. In the scope of Stemmatters and Accelerated Biosciences partnership, Stemmatters received 2 hTSC-derived iPSC clones to establish master cell banks, the clone 1 and the clone 6. Currently, research use only or dual master cell banks are being established, and in the future we plan to manufacture GMP master cell banks of human trophoblast stem cell-derived iPSCs. So cells have been taught and expanded for 3 subcultures to produce master cell banks of more than 100 million cells, to bank about 100 vials. And for the establishment of these master cell banks, cells has been maintained in normoxia. The growth media used is an animal component-free, so an HCF-free iPS culture media. The coating flask surface has been performed with a defined xeno-free matrix. We use a non-enzymatic clamping passaging, and cryopreservation of cells is made in a serum-free ACF cryopreservation media. Regarding the characterization of expanded cells, both iPSC clones from Accelerated Bioscience present normal growth with good colony morphology. We observe colonies with round borders, defined edge, compact cells, and cells also exhibit a high nucleocytoplasm ratio. Regarding the pluripotency markers expression, we tested the banked cells for 2x2 combinations of nano, OCT34, and SOX2. We carried out flow cytometry analysis of 3 these markers combinations and detected that more than 90% of iPSCs from clone 1 and clone 6 were double positive for the 3 combinations tested. This result confirms the highly pluripotent status of these cells. The direct differentiation of iPSCs in 3 germ layers, followed by a flow cytometry analysis of 2 markers for each germ layer, show that these cells have positive markers for endoderm, ectoderm, and mesoderm. And this demonstrates that these iPSCs are pluripotent and have the potential to differentiate into the majority cell types in the human body. In addition to demonstrating potency and lineage differentiation ability in these clones, as shown in previous slide, the banked cells exhibit a viability rate exceeding the minimum acceptance threshold. They also test negative for microbial contamination, and the endotoxin levels were below the maximum acceptable value. These results altogether confirm that the UO master cell banks were successfully established, with cells meeting the necessary quality standards for subsequent process development activities. In summary, Stemmatters is a CDMO qualified for GMP manufacturing of iPSCs master cell bank working cell banks, including iPSCs derived from trophoblast stem cell. The characterization of UO master cell banks established at Stemmatters demonstrates that both Accelerated Biosciences iPSC clones banked and characterized in our facilities met all evaluated quality control criteria. The cells exhibit high expression levels of pluripotency-associated markers, OCT34, SOX2, nano, consistent with an undifferentiated iPSC phenotype. Direct differentiation assays confirm that the hTSC-derived iPSCs retain the capacity to differentiate in the ectoderm, mesoderm, and endodermal lineages, demonstrating the 3 lineage differentiation potential of this cell. In the future, We will establish hTSC-derived iPSCs master cell banks GMP compliant, and we will also expand hTSC-derived iPSCs in 3D dynamic culture. Thank you for your attention, and I would be happy to share and to answer any question that you may have.